Hi class, so this is the video lecture for enzymes, and enzyme is the first topic for the final term. So what is an enzyme, or what is the function of enzyme, or why do we need to study enzyme? So the main function of enzyme is that they are biochemical catalysts, and so they are catalytic proteins, and again they are best known for their catalytic role. And almost every chemical reaction in the body is driven by an enzyme. So the important characteristic of an enzyme is that they are globular proteins. This is because of their tertiary structure. They are bent and folded into a spherical or globular shape. And because they are globular in structure, the hydrophilic part is at the outside or on the external part, while the hydrophobic are in the internal part of the enzyme. And so, they are soluble in water. So again, enzymes are biological catalysts that speed up the rate of the biochemical reaction. Now, in this presentation, you can see here a hill. So this hill here is what we call activation energy. So what is activation energy? This is the energy input required to initiate the reaction. So it is the input of energy before molecules will react together. And on the upper part, this is without the enzyme, and you can see the difference, right? If without the enzyme, we need a lot more of energy for the reaction to proceed. While with enzyme, yes, the activation energy is lowered. But enzymes are not consumed during the reaction, and so they are reusable. Again, the enzyme will lower the activation energy only. So again, catalysts are substances that speed up the rate of a chemical reaction, and enzyme cause cellular reaction to occur millions of times faster than corresponding uncatalyzed reactions. Enzyme can increase the rate of a reaction by a factor up to 10 to the power of 20 over uncatalyzed reaction. So we have here the activation energy. So in this activation energy, we have a delta G here, and this delta G here is the what we call the free energy change. And to get the delta G, we just have to subtract the final energy or final G to the initial G. Okay, and so class, you can see here that even with enzyme or catalyst and with even without catalyst, the delta G will not change. It is still the same. What changed again? What alters or what change in the if there is a catalyst is that the activation energy only they lower the activation energy. So the enzyme alters the rate of reaction but not its free energy change or position of equilibrium. Okay, so again, what are enzymes? They are biological catalysts that speed up the rate of biochemical reaction, and most enzymes are 3D globular proteins because of their tertiary and quaternary structure. So these are the important terms that you need to understand for the activity and biochemical nature of enzymes. So first, let's have active site and substrate. So what is an active site? So all enzyme molecules contain a special pocket or cleft called the active site. So in here, okay, so the catalytic activity of an enzyme is restricted to this small portion called the active site. So the substrate is the substance upon which the enzyme acts. So this substrate is the reactant in biochemical reaction. Again, the substrate is the reactant, and this is where the enzyme acts upon. So the first step in reaction catalyzed by an enzyme is the formation of a specific association between the molecules called the enzyme substrate complex. But this association between them is only temporary. So when in its proper dimensional shape, a 3D shape, enzyme has an indentation on one side of the structure, and this indentation is known as the active site. And it is lined with amino acid chains. So this active site is involved in catalysis, where the substrate or the substrates attached to is because of the non-covalent forces like hydrogen bonding, electrostatic attraction, hydrophobic interaction, just like the tertiary as in the tertiary structure. So what is the composition of enzyme? Most enzymes are large protein molecules with complex 3D shape, but we also have non-protein enzymes, and non-protein enzymes are few enzymes that are made up of RNA, and catalyze cellular reactions involving nucleic acid. And one example of a non-protein enzyme is a ribozyme. So ribozymes are enzymes that is made up of nucleic acids, and they are catalytic RNA involved in the removal of parts to form a functional RNA molecule. So we have a simple and globular enzymes. So simple, what we have discussed in our last topic, diba? Simple proteins only consist entirely of amino acid units. So the same with this one. A simple and globular enzyme, they only consist entirely of amino acid units. An example of this is pepsin, trypsin, and ribonuclease. But we also have conjugated or complex protein, and we call them holoenzyme. And in holoenzyme, there is a protein part, which we call apoenzyme, while the non-protein part, we call the cofactor or the coenzyme. So for you to imagine what is an holoenzyme, again, a holoenzyme is a conjugated or a complex enzyme, and it consists of apoenzyme. So the apoenzyme is the protein portion, but this apoenzyme is inactive. Okay? Kung siya lang inactive, so it needs a coenzyme or a cofactor. So what is the difference between a coenzyme or a cofactor? So a coenzyme is an organic compound or substance, while cofactor is the inorganic substance. So these two are the non-protein portion, and they are the activator. They activate the apoenzyme. So once they are attached to the apoenzyme, they are now called holoenzyme, and the enzyme is now activated. So the holoenzyme is the biochemically active biochemically active conjugated enzyme produced from this apoenzyme and a cofactor of coenzyme. So again, what is a cofactor? This is the inorganic substances. So when we say inorganic, these are the examples, the metals or the ions. So these are some of the examples of cofactors. So again, cofactors are the inorganic substances, and they attach to the um, um, duol sa active site. So for example, like this one, the apoenzyme here is unable to bind to this substrate, and when this cofactor, in this case, the copper 2 plus is available, it will bind to the apoenzyme. Now the active site is activated, or the holoenzyme is activated, and it has a correct configuration with the substrate, and so there will be an enzyme substrate complex form. Now, the next one is the coenzyme. So these coenzyme are small organic molecules acting as carrier molecule, and some or many of these are vitamins or provitamins. And we have two types, the co-substrate and the prosthetic group. Co-substrate, they binds transiently or loosely to the enzyme near the active site, while the prosthetic group will bind tightly or permanently to the active site. Okay? Or to the enzyme, I mean. And examples are for co-substrate, NAD+, while for the prosthetic group, it's 5. So here are some of the common coenzymes. You can see almost in these examples are vitamins like vitamin B1, B2, B3, B5, biotin, folic acid, ascorbic acid, vitamin C. So those are small organic molecules that are called coenzyme, and they activate the apoenzyme. So we have a situation here. So our collagen, it is a major structural material in our skin, in our blood vessels, in our tendons, ligaments, and this collagen contains hydroxylysine and hydroxylproline. So in the hydroxylation of this amino acid in collagen formation, they are catalyzed by an enzyme, and these require an ascorbic acid. So that enzyme, the apoenzyme, needs vitamin C, ascorbic acid, for it to catalyze this lysine and proline. And so if there is a deficiency, kulang of vitamin C ang ato ang body, the hydroxylation is impaired, and what will happen to the collagen, or to the structure of the collagen? So the triple helix of the collagen is not assembled properly. That is why ang ginaingon gid sa doctor, di ba? Or gina-suggest or gina-advise gid sa to ang we need vitamin C every day. This is one of the reasons. Now let's go to nomenclature or naming and also classification of enzyme. So for naming, so we can retain old traditional names. So the traditional names has a suffix "in". So these are still found in many of the digestive enzymes like pepsin and trypsin. Also, uh, they can be named from the species of origin. So kung aha sila nakita, aha sila um, na-discover. So like papain from papaya and papaya and ficin from ficus. Now, for the um, modern or sa ato nga generation, ang atong ginagamit now is the suffix ase. So this suffix ase identifies a substance as an enzyme like urease, sucrase, lipase, or they are all enzyme designations. So those who have ase nga suffix, we can say that those are enzymes. So enzymes are most commonly named by using a system that attempts to provide information about the function rather than the structure or to the type of reaction of the enzyme. So example, si oxidases, mga oxidases, they catalyzes oxidation reaction while hydrolases catalyzes hydrolysis reaction. So in number one class of naming, so the enzyme is generally named by adding ase to the end of the name of the substrate. Again, the suffix ase is added to the end of the name of the substrate. So the next is the identity of the substrate is often noted in addition to the type of reaction or function, like alcohol dehydrogenase. This enzyme oxidizes ethanol or alcohol. Okay, and there are also more examples there. And infrequently, the substrate but not the reaction type is given, like urease. So this urease is from the substrate urea. So this means that urease catalyzes the hydrolysis of urea. Lactase, this catalyzes the hydrolysis of lactose. Okay. So, we also, the same with our cars, dagan kaayo ta klase-klase nga sakyanan. So, what, unsay nakapa-special sila or unsay nakapahatag sa ila og identity is their plate number. So, the same with enzymes. We have a lot of enzymes, but we have a code. They have a code in order for them to be identified. And this code is from the International Union of Biochemistry and Molecular Biology. So they develop a nomenclature for enzyme. So each enzyme is described by a sequence of 4 numbers preceded by the EC. So this EC denotes as enzyme commission. So we have 4 numbers here, and the first number, it will tell us the class. Now, at second number will tell us the subclass, third, the sub-subclass, and the fourth is the serial number in that class. So, for the first number, or the class, or the classification, categorizes the enzyme based on its reaction. So, on the first number here, we have these 6 classes, major classes of enzymes. So, oxidoreductase, transferases, hydrolases, lyases, isomerases, and ligases. We will discuss this in details, on the next slides. Okay? And then the second and third number in this code, second and third, further define and narrow the details of the reaction type. They describe the reaction with respect to the compound, group, band, or product involved in the reaction. And the fourth number will have or will give us the order of the individual entries. So these 6 major classes, you have to memorize this one. Okay? And also, you need to identify unsay may mga things nga different sa ila, or when you will see the reaction, you will now can directly identify if it is oxidoreductases nga enzyme or lyases enzyme. Because in our level, sa ato nga course, diri lang ta mag-deal sa major class of enzymes. So we have an example here, CEC2711. So from there, the first number is 2, and so we can tell that the major class of this enzyme is a transferase. And then the second number, 7, the subclass, so it transfer of phosphate, and then 1 and 1. So this specific enzyme is a hexokinase, and it has this name. Mao gid ni ang ngalan sa enzyme. So there are a lot of examples here in this table. So these are the frequently quantitated enzymes. So unsa may common diri. For example, si pyruvate kinase. So si PK, na siya code nga 2714. So in here, mga trypsin. Si trypsin na siya 34214. So those are different common quantitative na mga enzymes. So let's go to the first major class of enzyme, the oxidoreductase. So this oxidoreductase, we can, to have a clue or a clue to look for, to know a reaction is catalyzed by this oxidoreductase is there is a loss of hydrogen. So from the word oxidoreductase, there is this catalyzes an oxidation and reduction reaction. So these are the different types of oxidoreductase. So first ones are oxidases. So from its word, oxidases, oxidation of a substrate. This involves in oxidizing or taking away electrons from a substrate. Another is reductases. So reduction, or reduces, or giving electrons to a substrate. So have you wondered while you are peeling or kanang mag-slice ka og apple or patatas or unsa pa na nga fruits, unsa man siya, mag-unsa iyang color for some time, di ba, mag-brown or mag-reddish? This is because of an enzyme reaction. So the enzyme involved is an oxidoreductase. Specifically, it is called a phenolase. So this phenolase is a conjugated enzyme and it has a copper in it. So when it is copper, unsa gani iyahang is it a cofactor or a coenzyme? Okay, so this phenolase has a copper in it. That is why kana daeng ma-oxidize ang ato ang mga fruits like apple, pear, patatas. That is because of this conjugated enzyme called phenolase. So we can prevent or slow this browning in several ways, like immersing our food or our fruits sa bugnaw nga tubig. So this will slow down the browning process. And also we can do we can pag atong ibutang sa refrigerator ang ato ang mga pagkaon or ato ang specifically fruits like apple o pear and a vegetable like potatoes. So this will slow down the enzyme activity of phenolase. Another oxidoreductase is the hydrogenase. So this is the removal or addition of hydrogen atoms. So class, so kung inani lang ang ihatag sa quiz sa exam, so dapat kabalo mo ay kani siya unsa ni siyang enzyme, unsa ni siya nga major class sa enzyme. So you have to identify unsa may common, unsa may or unsay difference between sa different na mga enzyme by just looking on their reaction. The next major class of enzyme is transferases. So in here, that there is a transfer of a functional group between a donor and acceptor. And the two major subtypes of transferases are transaminases and kinases. So kinases, there is a transfer of phosphate group between the substrates. So you can see here, the ATP, the phosphate group of the ATP will transfer to glucose, and so it will become a glucose 6 phosphate. This is because of the hexokinase enzyme. Another is transaminases. So the functional group na mag-transfer po saya is the amino group. So from the amino group of glutamic acid, it will transfer into pyruvic acid, and so it will have a product of alanine. So this is because of the transaminase called ALT, or the alanine transaminase. The next major class of enzyme is hydrolases. So this catalyze the hydrolysis of various bonds. So this will add water across a bond. So an enzyme that catalyzes a hydrolysis reaction in which the addition of water molecule to a bond causes the bond to break. So these hydrolysis reaction are central to the process of digestion. So as we already know, proteases. So these proteases are the enzymes or the catalysts. They catalyze the hydrolysis of the proteins. Okay? And they hydrolyze the peptide linkage or peptide bond. So, after hydrolysis, unsa gani ang product na ma-form or ma-product na makuha? Your amino acid. So, they need proteases in order to break the peptide linkage or bond of the protein. Now, for carbohydrates or sugars, they also need carbohydrases. So, this is the They catalyzes the hydrolysis of glycosidic bonds. So, for example, si alpha amylase and maltase, or the sucrase, lactase, cellulase, they are carbohydrases. Another one is lipases. So, these lipases hydrolyzes the ester linkage in the lipids. So, the lipids is what we call a TAG or triglyceride or triacylglyceride. So, once there is a lipase, it will undergo hydrolysis, and so it will become a glycerol and fatty acid. Another one is nucleases, hydrolysis of sugar, phosphate ester bonds in nucleic acid, and phosphatases, hydrolysis of phosphate ester bonds. The fourth one is lyases. So they cleave various bonds by means other than hydrolysis and oxidation. So these lyases will add water, ammonia, or carbon dioxide across double bond, or it will remove these molecules, this water, ammonia, or carbon dioxide to produce double bond. So example, so again, they catalyze the dissociation of a molecule without using water. So I type is dehydratase. So this is the removal of the components of water from a double bond. So you can see here, this will the blue ones will be removed, hydroxyl and one hydrogen, and will produce a double bond on the molecule. We also have the carboxylases under the lyases. So this is the removal of carbon dioxide from substrate, while diminases, removal of ammonia from substrate. Next, second to the last, the isomerases. So from the word itself, isomer, so isomerases, they catalyze isomerization changes within a single molecule. And so they carry out many kinds of isomerization, like from L form to D form, and also shift of chemical groups. So this will catalyze the conversion of substrate into another compound that is isomeric with it. Okay, diba? When there are two isomers, and when your compound has an isomer, it is a different compound. It is another compound. And so, when using with isomerases, when it will catalyze and form another isomerase, it is another compound. So, this will facilitate intramolecular rearrangement in which bonds are broken and form, or they can catalyze conformational changes like this one. From malate, malate has a cis configuration, but with malate isomerase, the malate will now have a trans configuration or trans nga form, but this trans nga form is now a different or another compound, and this is already a fumarate molecule or compound. So we have different types of isomerases. One is racemases. So this converts from D to L isomer or vice versa. Also mutases, transfer of a functional group within a molecule. So you can see there, the phosphate group is at the bottom part. However, using this mutase enzyme, it transfers from bottom to its side. We also have epimerases. This is the conversion of one sugar epimer into another. Okay, the last one, the ligases or the synthetases, they join two molecules with covalent bonds. So they catalyze reactions in which two chemical groups are joined or ligated with the use of energy from ATP. So another clue para makabalo mo nga, ay, kani siya nga class nga enzyme is ligase or synthetases, because all ligases and synthetases needs an ATP, or it uses energy from ATP. So kung ang ATP naa siya sa reactant nga side, possible nga ang kana nga class sa enzyme is class number 6 or ligases. So again, they catalyze ligation or joining of two substrate, and this requires chemical energy. So examples or types of ligases is synthetases, formation of a new bond between two substrate. So again, this needs a participation of ATP. So from glutamate, nahimo siya og glutamine. So this is a formation of a new bond of amine. Another is carboxylases, formation of new bond between substrate and carbon dioxide again with ATP on the reactant side. So we have a practice exercise here. So you can see already there is a loss of hydrogen from this substrate. Okay, so this is a oxidoreductase na class of enzyme. Another one, this is water in the reactant site, and so we can say, and also there is a breakage, right? This is a peptide, dipeptide, and there is a breakage, and so we will have an product of amino acid. And so this is hydrolase. Class of enzyme is hydrolase. So this is just a summary of all the major classes of enzyme. Again, you have to memorize all of this or familiarize, especially kung tagaan lang mo og chemical equation or reaction, dapat makabalo mo unsa na nga class sa enzyme ang ga-undergo ana nga reaction. Now, let's go to the mechanism of enzyme action. So, there are characteristic features of enzymes, and first is the catalytic power. So, in the catalytic power, the activation energy and the mechanism of action. So, for the mechanism of enzyme action, again, formation of an enzyme substrate complex as an intermediate species provides an alternative pathway with lower activation energy through which a reaction occurs. So the enzyme substrate complex can lower the activation energy by bending substrate molecules in a way that facilitates bond breaking, helping to reach the transition state. So you can see here, class, in this activation energy chart, the red one is for the activation energy with catalyst or with enzyme, while the blue one is without the enzyme. And so you can see there that there is a lower activation energy if there is a presence of a catalyst or an enzyme. The next one is its specificity. So this is the selectivity of enzyme to their substrate. So we have a theory here or a model here that deals about the specificity of the enzyme. So first is the lock and key theory. So this is postulated by Emil Fischer in 1894. So in here, lock and key theory, just like our lock and our key. So the key, which is the substrate or the reactant that fits into the keyhole, which is our active site of the enzyme, which is our lock, will only kung unsa lang ang correct nga shape sa ato ang lock o kung correct pod ang shape sa substrate, sila lang ang mag-bind together. So the active site in the enzyme has a fixed, rigid geometrical conformation or shape which is complementary to the substrate. So only a substrate with a matching shape can fit into it. So that is for lock and key. But because of its sa iyang kastrikto sa iyang model, we have now an induced fit theory. So this model is suggested by Koshland in 1958. So these models allow for small changes in the shape or geometry of the active site of an enzyme to accommodate a substrate. So this is more thorough explanation for the active site properties of an enzyme because it includes the specificity of the lock and key model coupled with the flexibility of the enzyme protein. So, there is in here, dili pareha sa lock and key nga dili gid mag-change ang specific ra gyud nga substrate ang maka-attach sa active site, but for induced fit model, there can be a small change on the active site to accommodate the substrate. Now, the force that draws the substrate into the active site are the same forces that maintain the tertiary structure or the folding of peptides. So, again, these are the three interaction between the substrate and the active site, the H bonding or the polar-polar interaction, the hydrophobic interaction between nonpolars, and then the salt bridge or the electrostatic interaction. Now, the specificity is the ability of an enzyme to choose exact substrate from a group of similar chemical molecules. And this specificity is a molecular recognition mechanism, and it operates through the structural and conformational complementarity between enzymes and substrates. So, this characteristic of enzyme class is very important because there are a lot of enzymes and there are a lot of substrates. And so, because of its specificity, naalangyoy unsa nga sa kadaghan substrates, naalangyoy exact or nalanggi pipila lang ka substrate nga maka-attach sa the active site sa enzyme nga maura pod gyud iyang gikinahanglan on that reaction. Okay? So, this is the end of the part 1 of the video, lecture video. So, the next part will start at the different levels of specificity of enzyme. Okay?